Hello, everybody, and welcome to this Nature Research Academy's webinar in systematic review and meta-analysis delivered in partnership with the Egyptian Knowledge Bank. My name is Hiba, and I'm going to be facilitating this webinar. I will be helping the trainer, Dr. Jeffrey Robbins, to run the polls and exercises, and I will be sharing the Q&A sessions. I will also be helping anyone with technical difficulties. Before I hand over to your trainer to get started, I will first show you how to use GoToWebinar. I will explain how to navigate the dashboard and how you'll be able to interact with us during the webinar. As you can see on your screens now, please have the dashboard maximized by clicking the orange arrow at the top of the interface. As part of some of the exercises, we will ask you to raise your hand. The bottom of the picture of the hand allows you to do so. Finally, at the bottom of the interface, there is a question box. Please type to us here if you are having any technical difficulties. If you have any problems during the webinar, we recommend logging out and logging back in. You can use the question box for questions or to respond to exercises. There will be a Q&A session in the middle of the webinar and the final session at the end. So please submit your questions as soon as you think of them, and we will answer as many as we have time for. Your question and comments will only be seen by us, so they are private. We will also be asking you to vote in some polls today. These will appear on your screen, and you will be able to vote just by clicking on the screen. Great, so that covers everything from my side. I will now hand over to your trainer, Dr. Jeffrey Robbins.
Thank you very much, Eba. And I would like to say good morning to everybody who's joining us. We're happy to have you with us here for these two webinars focused on systematic reviews and meta-analyses. Now, before I begin, one thing I would really like to emphasize is that we're not going to go through a lot of the statistical aspects of how you conduct these systematic reviews, or in this case, meta-analyses. But really what we're trying to emphasize is from an editor's perspective. What is it that editors and reviewers are looking for when you're preparing and communicating the systematic reviews and meta-analyses that you've done? Now, if you want more detailed information about how you should be conducting them, how to actually be doing the search strategies, how to do the statistical syntheses, I encourage you to reach out to your librarian, reach out to any friends you have that are statisticians, epidemiologists, and they'll be able to help you with a lot of those more detailed questions or concerns that you have. So we're really coming at this more from an editorial perspective to help you publish your systematic reviews and meta-analyses. Now, in that regard, we are going to be dividing this one workshop across two separate webinars because we don't want you sitting in a chair for 4 hours listening to me speak to it, watching me on your screen. On the first webinar, the one we're going to do now, we're going to be talking about identifying the right studies, how do you communicate that in your papers, and also the data extraction you should be using that are the international standards that, again, you are expected to be familiar with. In the second webinar, we're going to be talking about biases, so biases within the studies you're evaluating, but also your own biases in the systematic reviews. So how do you rate the quality of that evidence and avoid that type of meta bias? And then at the end, we'll talk about doing the qualitative synthesis for a systematic review or the quantitative synthesis for a meta-analysis. Now, I would like to emphasize that in the handout section of the GoToWebinar dashboard, you will see both the participant guide, which I hope you've already had a chance to download, but also you will see the slides from this first webinar. We will update the slides with the next slide deck when we begin the next one. Now, in the participant guide, I would like to highlight some valuable information. Firstly, on page 3, you will see the agenda for these two webinars, so the information that we will be covering. On page 7, you will see 4 through 7, you will see some of the key points that will hopefully be a useful reference for all of you. On pages 8 through 11, we do have the activities that we are going to go through today. This is meant to be an interactive workshop. And lastly, on page 12, we have some additional resources you can find online if you want more detailed information about conducting systematic reviews and meta-analyses. Now, let me tell you a little bit about myself. So my name is Jeffrey Robins, and I'm Editorial Development Manager for Nature Research. Now, I received my PhD in pharmacology from the University of Pennsylvania in the United States. Upon receiving my degree, I then decided to leave the US, and actually I moved to Asia. So I've been living in Asia now for over 18 years. I'm currently based in our Nature Japan office here in Tokyo. Now, before I joined Nature, I did conduct research at institutes in Singapore as well as here in Japan. In this time, I've published a number of international articles. I was actively involved as a peer reviewer for a number of international journals. But then about 9 years ago, I decided to leave research, and I actually joined first an academic editing company where I edited the manuscripts, predominantly clinical or biomedical in nature, to help authors improve the scientific message in their papers before they send them to their target journals. And then about 5.5 years ago, I joined Nature as editorial development manager, where one of my key responsibilities is developing and conducting training workshops for editors, for peer reviewers, and for researchers, in the end to hopefully improve the impact of the published literature. Now, although I work for Nature, we are part of a much larger company called Springer Nature. So Nature themselves really don't publish systematic reviews or meta-analyses, with the exception of scientific reports, but the Nature journals generally do not. However, in our Springer journals and BMC journals, we do publish a lot of systematic reviews and meta-analyses in those clinical journals. And so what I'm going to be communicating is from the perspective of those editors, those reviewers. What is it that they're looking for? What is it that they are going to be expecting from you when they are evaluating your manuscripts, when you are discussing the analyses you've conducted? Now, what are we doing here in Egypt? Well, so Springer Nature does have a large multi-year partnership with the Egyptian government, where we're providing content of all the journals, all the books that we publish, freely accessible for you. We're also actively publishing over 30 Egyptian journals as part of our portfolio to try to improve the international exposure of Egyptian research worldwide. We also providing editing services to help to improve the language in your manuscripts. And as we're doing here today, we are providing training for Egyptian researchers, trying to raise the level of Egyptian research to be much more competitive on a global scale. And as Heba mentioned earlier, all of this is done in collaboration with the Egyptian Knowledge Bank. Now, if you haven't done workshops with us before, these are meant to be interactive. We don't want you just sitting back, watching, listening. You're going to get bored. So we are going to ask you to get involved with us. And we try to have variety of how you'll have a chance to interact with us, as are indicated by these icons on the screen. So when you see the red icon, you know we are going to launch a poll. And in that poll, you'll be able to vote for an answer, or more than one answer in many cases. When you see that blue hand, you know we're going to be asking you to raise your hand in response to a question or a comment that I've made. Can we try that out now? Can everyone please raise their hand for me? Can you press that hand button on your GoToWebinar dashboard? Fantastic. I see close to, now close to 50% of you have raised your hand, means hopefully you've been able to identify it. So when I do ask you to raise your hand later, it should be easy for you to find that button. And then lastly, when you do see that purple question mark, you know I'm going to be asking you to write in your answer to the questions box. And so hopefully by having a little variety of how you interact with us, will make this webinar a lot more engaging and interesting for all of you. So with that, let's now move into the content of this first webinar. Now, in this first webinar, we're going to be covering 3 important topics. First, highlighting the importance of systematic reviews and identifying the right research questions. Without that, you will never be able to publish it. We'll then move on to talk about how do you search for studies and recommendations of search strategies and databases to consider. And then lastly, at the end of the webinar, about extracting and analyzing your data. So let's go on with the first topic of the day, which is the importance of systematic reviews. What are their benefits for the field? So with that, we're now going to go into our first poll. Why do you think systematic reviews are important? So Heba, can we launch poll number 1 for everybody?
Thank you so much, Dr. Jeff. So everyone, now you should see the poll on your screen. So please choose one or more of the 4 options and submit your vote to us. And I will keep the poll open for a minute to give you time to vote. So what do you think? Why are systematic reviews important? Is it because they are valuable in evidence-based medicine, or because they are objectively synthesized, published and unpublished studies, or because they help establish my reputation in the field, or that they are any different from literature reviews? Thank you so much, everyone, for your participation. So I will now close the poll and I'm going to share the results on the screen.
Okay, fantastic. So I can see 75% of you selected that they are valuable for evidence-based medicine, and that is definitely going to be the case. We'll talk a little bit more about why they're so valuable. So I'm happy to see the majority of you do already understand that. And of course, what makes them very unique and different from literature reviews is they are much more objective, and they do synthesize both published and unpublished studies. And that's really what distinguishes them from a basic literature review. Now, I do see a third of you said it'll help you to establish your reputation, potentially. If you write a good systematic review or meta-analysis that's having an important impact in the field, of course, that'd be a great way for you to help to establish your reputation. And then it is, so I do see about 1/5 of you selected, are they any different from literature reviews? And yes, they are. They're very different from literature reviews. So thank you very much, Heba. So there is sometimes some confusion about a systematic review and a literature review. And often after I do these types of workshops, people often tell me, Jeff, doing a systematic review is actually really difficult and really challenging. It's not easy to do. So when we're talking about systematic reviews, it's much more focused than a literature review. Literature review you can see more as a broad overview of a topic. A systematic review is that it is systematic. So in terms of goals, so a systematic review has a very defined research question, just like any primary research study would have. Literature review, on the other hand, is just a summary of a basic topic in the field. In terms of authors, one person can write a literature review. Generally, you need a minimum of 3 authors to conduct a systematic review. And we'll talk more about the importance of having independent reviewers when conducting the analysis later. In terms of timelines, literature reviews can be done quite quickly, often in weeks, maybe even up to a month or 2. Systematic reviews in general take about a year to year and a half for most authors. They are very long and they are time consuming. In terms of searching, literature review is just based on the published literature. It's what that author knows and is familiar with in papers that have been published. A systematic review should evaluate both published and unpublished studies, which is not going to be discussed in a literature review. And the value, as we highlighted in the poll, really systematic reviews bring a lot of weight to evidence-based medicine to help clarify the efficacy of a certain intervention for a condition of interest, for example, while literature review is more just an overview of the literature that author is particularly aware of. So where did systematic reviews come from? Well, the first systematic review was actually 1955, which was trying to evaluate the effect of the placebo, who they found was to be effective in about 1/3 of patients. 1976, Dr. Glass, who was a psychologist, was the first person to coin the term meta-analysis, which we now use routinely when you're talking about the statistical synthesis. And then in 1992, the Cochrane collaboration was established, which really tried to emphasize the importance of systematic reviews and meta-analyses in clinical practice. Now, the Cochrane collaboration is named after this British epidemiologist named Archie Cochrane. And he was someone who really emphasized the importance of randomized controlled trials for better informed healthcare, and because of that is often seen as the father of evidence-based medicine. So you can see the tight association that the Cochrane collaboration has with systematic reviews with evidence-based medicine, which is why they took Archie Cochrane's name. Now, if you're not familiar with the Cochrane collaboration, I really encourage you to visit their website. They have a wealth of information, including a freely available handbook. It is incredibly long, but it's available online, and it's a great resource. I often use it and refer back to it from time to time, and I put the URL here. So if you ever want additional, really more in-depth information, this is a great place to go. Now, because they do understand the training that needs to be involved, they do have a very in-depth online training platform. They go into a lot more information than we can in these 4 hours. I do want to emphasize it is not free. You can see it does cost £200, so it's not something you can freely do. I also want to emphasize they have no association with us whatsoever. They're actually owned by another publisher, Wiley. So I'm not recommending them because they have any financial interest with us. In fact, it's with one of our competing, our main competitors. I simply encouraging you to refer to them because I think it is a really excellent resource for systematic reviews and meta-analyses. So where do systematic reviews and meta-analyses stand in terms of clinical relevance for the field? Many of you may be familiar with this pyramid that's often used to discuss the levels of clinical relevance. It's never to say that one study design is better than another, but in terms of the value that it's bringing to active clinicians will vary. So at the bottom of the pyramid, we have case reports looking at the unique presentation of an individual patient. And then at the top of the pyramid, we do have systematic reviews and meta-analyses. Now, the reason of this is because of their synthesis. They're synthesizing together generally RCTs, but sometimes observational studies. And by providing this objective systematic synthesis, really provides that extra layer of value. But I also want to emphasize, of all the clinical study designs, systematic reviews are the only ones that don't accumulate primary data. They are using the data that's already available, therefore it's called secondary data, and is now synthesizing them either in a qualitative or a quantitative manner. So because of that, when we're talking about the units of analysis, for clinical studies, all the primary data, it's all the units of analyses will always be the patients or the participants of those studies. Well, for systematic reviews, that's not the case. The units of analyses are the studies themselves. And although I do have them here as papers, these not necessarily meant to be published studies, because as we'll discuss, that you should be trying to identify both published and unpublished studies. Now, because of that, because the studies themselves are the units of analyses, you also have to be aware that the strengths or the rigor of those studies will differ. Some studies will be very large and therefore will have a lot of strong power and will be a lot more useful. Smaller studies will have a lot higher variance and therefore often could be less useful, may even be underpowered for the field. And so what you'll do, and we'll talk about this at the end of the day, is you're going to be assigning weights to those different studies based on the variances that you're going to be observing and evaluating when you're assessing those units of analyses. So generally what a systematic review is, you've heard me use the word synthesis several times. That's essentially what a systematic review is. It is a synthesis of the already established evidence from pre-established or pre-specified eligibility criteria about a specific question. In other words, everything is a priori. It is something you plan for in advance. And generally, not always, generally it's about the efficacy of a treatment. So what type of synthesis are you going to do? Well, it depends if you're doing a systematic review, which is a qualitative synthesis, or you're going to do a quantitative reevaluation, a statistical synthesis, which would be termed as a meta-analysis. All systematic reviews have a qualitative synthesis. Some systematic reviews also have a meta-analysis. I want to emphasize that you don't always have to do a meta-analysis. You should only do so when it makes sense. And we'll be talking about that a bit later in this workshop. So why would you want to conduct a systematic review? If you're not the one actually obtaining the primary data, well, you may think, well, in that case, it makes my life much easier. If somebody else already collected all of that data and all I need to do is synthesize it, well, that makes my job much easier. Who agrees with this statement? Who thinks that a good reason to do systematic or disagree with this statement? Who thinks that just because other people obtain the primary data, that makes doing a systematic review much easier? I can see, okay, not that many, about one-fifth of you raising your hands, but that means about 4/5 say, Jeff, this statement to me makes a lot of sense. Well, again, let me emphasize, systematic reviews are often, especially for first-time people, a lot more challenging than what they would have expected. Good systematic reviews, and we'll talk about how you develop a good, technically rigorous systematic review in this workshop, will take you many months. On average, they take most people about a year and a half. If you work really hard and you have a lot of time dedicated to it, you maybe be able to get it in half that time, maybe 9 months, but often they can be drawn out to 2 years or maybe even more. So, but why should you do so then? Well, again, you've identified here's an important question that needs to be asked, needs to be answered, and going to have strong relevance for the field. And you've already seen that many studies have been conducted already to try to address it, but the results of those studies is not conclusive. Some say it looks beneficial, some says it doesn't show any conclusive benefits, and therefore, from a clinician's perspective, it's really unsure whether or not that intervention is going to be useful. And so the whole point then is, should we continue conducting additional RCTs, or based on the available evidence we already have after we do their synthesis, can we conclude that this intervention is going to have efficacy or not? So for example, here's an older meta-analysis published a few years ago, and they were looking at the clinical efficacy of ACE inhibitors for non-diabetic renal failure. Now you can see from the actual forest plot, 10 studies have been conducted already. But when we look at the whether or not it did have a favored ACE inhibition, what you can see is that none of these studies, one was probably a bit closer than the others, but there's no conclusive evidence supporting the use of ACE inhibitors. But when a meta-analysis was done, they could validate that yes, ACE inhibitors were going to have a protective or be useful for non-diabetic renal failure. And this is something we would not be able to assess from the individual studies alone. And I think this really is, when we were talking about evidence-based medicine, this gives clinicians the evidence necessary to make the decisions in the clinic. So now, in terms of the fact that you're not collecting the primary data yourselves, can actually have a really strong value for you as a busy clinician as well. First of all, because it's not a longitudinal study, you have to follow participants and you have scheduled follow-ups throughout the actual study design. That means you can go in and out whenever you have time. When you have time, you can go back to start reanalyzing the data you abstracted. When you're busy, you set it aside and then you do your other work. So there's a lot more flexibility than what you could have with an RCT or cohort or a case-controlled study, for example. Because you're not conducting the study itself, it's more just of analysis. It's a lot of sitting down at the computer and analyzing all of those papers or all of those studies. They're a lot more affordable. And when funding can be an issue, affordability is always important. And then I think another really valuable aspect of systematic reviews is, and a key aspect, as we'll talk about later, is evaluating the quality of the evidence, evaluating and assessing the biases in those studies. And by being familiar with the types of biases that plague these RCTs or cohort studies, when you're conducting your own, you can be more familiar and be more careful to avoid those types of issues in your research. Therefore, you're actually improving your skills as a clinical researcher as well. So I do think that there is a lot of really benefits and value for you to consider. Now, should you do a systematic review or meta-analysis? Well, some of the benefits of systematic reviews, they can be done for heterogeneous studies, so you can compare apples and oranges. You can't do that with a meta-analysis. But if you have very heterogeneous studies, you can do a narrative synthesis, so qualitative synthesis. And of course, as I mentioned, they are affordable. However, because it is a qualitative synthesis, it's a bit more subjective in how you're making those analyses. You're not making the analyses based on quantitative information, but rather your overview, your assessment of that information. And therefore, there can be a higher risk of bias than what you'd see in a meta-analysis. So here's an example of a systematic review, and I just want to emphasize how the data is presented in a typical systematic review that does not include a meta-analysis. This one, they're looking at the efficacy, effectiveness, and safety of vaccination against HPV in males. And so here's the data. This being presented, they assessed these are the different studies that they looked at, and they're presenting all the information related to those studies. And then in the text, they review that information. And we'll discuss more in the second webinar, a good ways that you should be conducting the actual qualitative synthesis and describing that information in your paper. Now let's compare that with a meta-analysis. One of the advantages, it is a lot more objective because it is a statistical reevaluation of that data, not a qualitative reevaluation. And it can increase the statistical power by pooling samples together, and therefore gives us greater confidence in the conclusions that can be made compared with the systematic review. Then also because of this, they're usually really at the tip of that hierarchical pyramid in terms of clinical relevance, but they're still quite affordable, although they do take a lot more time. Disadvantages is they do take a lot more time. It is a lot more involved than doing a typical standard systematic review. And gathering and extracting and evaluating that data can be quite challenging. You will need to work very closely with someone who's experienced in doing meta-analyses or a statistician to make sure you don't make any very common errors. And then the other key issues about meta-analyses, if you do have a lot of heterogeneity across the studies, it's not appropriate to do one. You will have to do a qualitative synthesis only. So it's only when that variance or the differences that you see across the different studies, you feel does make sense to pool that data together, can you actually do a meta-analysis? So there's a lot more limitations than what you would see for a typical systematic review. Now, in terms of data presentation, often the data will be presented not only as a table, but also importantly in a qualitative perspective as a forest plot. And this is what a lot of you are probably familiar with seeing. So in this case, when they're looking at the association of this polymorphism, what they had seen when the previous 6 studies, only 2 of those previous studies actually showed a strong correlation, a conclusive correlation. The other 4 was not clear. However, when they conducted the meta-analysis, they can see that yes, they can confirm that this association between this polymorphism and the condition was in fact going to be true. And that lets us know, do we need to conduct more studies? No, it's already conclusive, and therefore it's valuable for the field. So that's a brief overview of the introduction. Now let's go more into the strategies of how you conduct them properly. There are a lot of steps involved. First step is you have to write your research question. Again, it needs to be defined a priori what it is you're trying to evaluate. Then in purple, you have to identify the previous studies, both published and unpublished. In gray, once you've identified the appropriate studies that meet your inclusion and the exclusion criteria, you will then assess the quality of evidence to be able to assign weights for those different studies. Then you can go through and then summarize the evidences either as a forest plot for systematic review or summary finding tables for, forest plot for meta-analysis and the summary of finding tables for your systematic reviews. And then you'll have to interpret your results in your discussion and really emphasize the strengths, limitations, and the value that the systematic review or meta-analysis had for the field. Now, in terms of writing the research question, which is where we need to start, a good way to do so is with PICOS and FINER. Now, that now brings us to our second poll of the day. Who here is already familiar with PICOS and FINER? If you have attended our clinical research methodology workshop already, you're probably familiar with these acronyms, but let's first get an overview from everyone that's attending. So Heba, can we please launch poll number 2 for everyone?
Thank you so much, Dr. Jeff. So everyone, now you should see the poll on your screen. So please choose one or more of the 5 options and submit your vote to us. And I will keep the poll open for a minute to give you time to vote. So who is familiar with PICO and FINER? I have never heard of either. I know PICOs but not FINER. I know FINER but not PICOs. I know both of them. Is PICOs related to PICO? Thank you so much, everyone, for your participation. So I will now close the poll and I'm going to share the results on the screen.
Okay, so I can see the majority of you said you haven't heard of either acronym, which is fine. I'm always happy to introduce new information that would be beneficial for you. About a quarter of you are familiar with PICOS, but not FINER. Very few of you know FINER, but not PICOS. Not surprisingly, FINER is a lot less used than PICOS is. 15% know both. Maybe some of you who have attended our clinical research methodology workshops where we've discussed these in more detail. And then some of you ask, is PICOS related to PICO? And the answer to that question is yes, it is. So thank you very much, Heba. So these are basically acronyms to help you to refine a research question that you think is going to be valuable for your study. Doesn't matter if it's a primary research study or a systematic review and meta-analysis. So PICOS, it is an acronym, so each letter stands for something. P stands for population, so who is this systematic review going to be relevant for? I is the intervention, which treatment are you want to look at, or it could be an exposure you may want to evaluate, a prognostic factor as well, for example. C stands for comparison. What are you going to compare this with? O is the outcome. What is going to be measured? And then S is what's going to be really specific for a systematic review is the study design. So what kind of studies are you going to include? Only RCTs, observational studies, etc. So you need to make sure all of these should be clearly described in your study protocol before you even begin. Now, once you have your research question defined, then you can move on to use the next acronym, which is FINER. F stands for feasibility. Do you actually have the expertise and the ability to conduct the systematic review? It is very time consuming. Do you have time to spend over the next 1 or 2 years to really thoroughly conduct it? If not, maybe it's not an appropriate question for you to address. I stands for interesting, so should be an important question for the field right now, something that's relevant. N is novel, so has this already been done? Or is this something really new? Is it a derivative type systematic review? Maybe something similar had been done before, but you're trying to evaluate different exposures or maybe different routes of administration or a different population, for example. Ethical, in terms of systematic review, what we really mean are the ethical guidelines you should be following to ensure that you're not going to be wasting time, you're providing evidence that will be valuable for the field. So we'll talk about Prospero registrations and PRISMA checklists soon. And then R stands for relevancy. So is this study that you're conducting relevant? And if so, for whom? Patients, clinicians, administrators, or a combination thereof. So let's look at an example where the research question of a systematic review was very carefully being described in the paper. So in this case, we're looking at prognosis, a prognostic value of receptor status conversion following neoadjuvant chemotherapy in breast cancer patients. So first, what they do in their paper, they introduce the topic that neoadjuvant chemotherapy is widely regarded as an important treatment option for early stage breast cancer patients and is able to achieve similar long-term clinical outcomes as adjuvant treatment. So they introduce the topic, now they define the outcomes of interest. Recent studies have reported that hormone receptor and HER2 discordance levels before and after neoadjuvant chemotherapy treatment. Then they talk about the problem. The results from several studies concerning the prognostic value of the changes in these receptor levels following neoadjuvant chemotherapy remains controversial. So yes, a lot of studies have been done, but we don't have any conclusive evidence one way or the other. And that's the motivation behind this study. So in this investigation, we conducted a meta-analysis to clarify if hormone receptor and HER2 discordance prognostic value and to explore the potential clinical value for these breast cancer patients. So in this case, we have a very clear description of the topic. What are the outcomes? Why is this an important problem for the field? What is the novelty or the originality of this research? And what is it they're hoping to accomplish? Now, once you have a general idea of the research question of what you're trying to explore, the next step is to register that in an online registry. Some of you may be familiar with clinicaltrials.gov or some of the other clinical trial registries. There are also registries for systematic reviews, and the one that's predominantly used is Prospera, which is based at University of York in the UK. Now, why do you think it's relevant? Why do you think it's useful to register your systematic review before you begin the study? Can someone please write into the questions box, what is the value there of doing this? There's no right or wrong answer. There can be a number of reasons why you think it's valuable. Any ideas? Okay, so one person said to avoid redundancy. Another said to check novelty, to keep the novelty of my ideas, to assess novelty. It seems like novelty or maybe intellectual property or avoiding repetition seems to be the most important reasons why people think to do so. And that's definitely the case. And it's useful. So what you do, for example, before you even consider registering, see what other people have already done. So for example, if I put in hepatocellular carcinoma and I can see there's 246 records, you can see if the review is ongoing or if it's already completed. If a review is ongoing, should you do it yourself? No. But what you can do is you can look at their study protocol. What are their outcomes? What are their patients? What are their interventions? And then you may be able to refine yours so you're not going to be duplicating those efforts. Now, if you don't register yours, then people can't avoid duplication of their systematic reviews either. So it's useful for you to register yours once you've decided this is what you're going to do. But again, as many people mention, you always want to avoid duplication. Always review the Prospera database when you're designing your systematic review to ensure it has not yet been done before. And if it has, well, how can you revise yours to improve its novelty or the relevance it has for the field? Now, some of you, of course, probably familiar with Cochrane reviews. Cochrane systematic reviews are conducted a bit differently, and it is a lot more in depth, but because of that, their standards are thought to be really high. First is what you do is you identify your topic and you establish the title of your systematic review, which should include the patient population, intervention, and outcomes of interest. At that point, you want to register that with the Cochrane collaboration, and that'll be with the specified Cochrane review group, of which they have several for you to choose from. So what you do is you will then write your proposal. You'll be submitted to the Cochrane Review Group, who will then do basically a peer review of it to say, yes, this is something important to do, or no, this is not something you should do. If it's been approved that, yes, this is going to be relevant after maybe some revisions, then they're going to publish the proposal in the Cochrane database, and therefore people already know it's undergoing. Once that's been approved, you then conduct the review as you normally would for systematic review and meta-analysis, write the final manuscript, you send it back to the Cochrane collaboration, where it'll then be re-reviewed, the final manuscript, you have to revise it, and then finally published. And you will be expected to update those Cochrane reviews over time as new evidence comes out. So it is a lot more involved. However, that's why Cochrane systematic reviews are often seen to be a whole nother level than the systematic reviews published in typical journals. Now, the other thing I would like to highlight are the PRISMA guidelines. These are guidelines, these are the international standards of how to conduct a good systematic review and meta-analysis. They do have a good checklist, flow diagrams, etc. And so if you're unfamiliar with systematic reviews, it's a great place to be familiar with what you're expected to do. And because of the issues of people not having that strong technical rigor in systematic reviews, some journals actually require that you complete the PRISMA checklist and a flow diagram in your actual manuscript. So here's, for example, one from JCI, that you must provide a completed PRISMA checklist. That way the editor feels confident you were adhering to those international standards. And the checklist has information about the title, abstract, introduction, your methodology, the results, discussion, and funding. So this is all the information, is 27 items you are expected to check off. Even if the journal doesn't require it, it's still going to be essential. I would recommend you to do so because it makes sure you are adhering to these international standards. Now, there's another guidelines which is specifically catered for observational studies. So if you're not going to be using RCTs in your systematic review, and instead you're going to be using, for example, cohort or case-controlled studies, you may want to use MOOSE, which is a meta-analysis of observational studies in epidemiology. And so, and I put the reference here, and you can go ahead and download that checklist, which again, will guide you through a lot of the information you are expected to be able to include in your systematic review or meta-analysis of those studies. So with that, let's now move into our first activity before we have our break. And this is going to be found on page 9 of your participant guide. Now, in this activity, we're giving you a scenario here. We say that two orthopedists discuss the recent advances in treating osteoarthritis of the ankle. And among the well-known studies in the field, which one should guide their decision-making in terms of the clinical practice? Is it A, the conclusions of a recent randomized clinical trial published in 2018 and performed on 70 Egyptian patients? B, now this should say literature review, not a systematic review. That was a typo in your participant guide. I do apologize for that mistake. So it's the latest literature review from a very famous orthopedic surgeon, which was published back in 2011. Or C, the conclusions of a Cochrane meta-analysis published in 2017, which summarized 6 randomized controlled trials of 240 patients. So which of these 3 studies, A, B, or C, would you recommend that these orthopedists should be using to guide their clinical practice? So Heba, can we please launch poll number 3 for everyone?
Thank you so much, Dr. Jeff. So everyone, now you should see the poll on your screen. So please choose one or more of the 3 options and submit your vote to us. And I will keep the poll open for a minute to give you time to vote. So which would you recommend? Thank you so much, everyone, for your participation. So I will now close the poll and I'm going to share the results on the screen.
So I'm happy to see that the majority, 80% of you, chose the 2017 Cochrane Review, which is synthesizing 6 RCTs. So we'll go through them. Some of the, now I'm not saying the other ones are not useful. However, you have to keep in mind the limitations as well. Thank you very much, Heba. So I would agree with the majority. Yes, C would be the best option to choose, to be the best recommendation to these orthopedists. Now for A, I'm not saying it's a bad study. Of course, it can be very useful. RCTs are considered to be a gold standard. It is the most recent study. It is related to the population of interest for an Egyptian population. So all of those are going to be very strong strengths and merits of the study. However, RCTs will have lower clinical relevance than systematic reviews or meta-analyses, keeping in mind this is only one study and it's only of 70 patients. So for B, so looking at this later literature review, and now the benefit of this, it is going to be a very reputable, famous author. So maybe someone you trust their opinions, and that's going to be a strength of this paper. However, it is also the oldest study. There's going to be a high risk of bias, both in publishing bias as well as subjective analyses of that information. And keeping in mind, literature reviews are just a narrative overview of where the field currently is now. And being this is from 2011, it's not very up to date anyway. And so for C, of course, Cochrane meta-analyses will have the highest clinical relevance. I would say of meta-analyses, Cochrane meta-analyses are really at the top of that pyramid, because they do adhere to these very strict Cochrane standards. There are 2 rounds of peer review. The first is the proposal, second is the actual study itself. They are updated as new evidence is made available. All of this really improves the benefits that they bring to the field. And because this is a meta-analysis, it is also a quantitative reevaluation, therefore this statistical synthesis, which helps to minimize some of the biases you may see in a typical systematic review. So with that, that now brings us to our first break of the day. If this is your first time joining a workshop with us at the Nature Research Academies, the way we organize these breaks is the first 10 minutes is for you. Please get up from your chairs, stretch, move around, maybe go have some coffee or tea, and then think of some questions you'd like to ask, and then put them into the questions box. Because when we come back in 10 minutes, we'll then go through the questions that you have, read them out anonymously, and go through them together. So with that, thank you so much for your participation so far this morning. Have a great break, and we'll see you back here shortly.
Welcome back, everybody. I hope you have. Hello, welcome back, Dr. Jeff. Welcome back, everybody. I hope you have enjoyed the little break. So we are now going for a Q&A session. So please allow me to read some of the questions that we got here, Dr. Jeff. The first question is the systematic reviews is just only for the medicine researchers?
Good question. Actually, we had several questions come in from different participants related to whether or not this is only for medicine, is it applicable for other fields, nursing, etc. And of course, it is applicable across all fields. Where is it most commonly done? Medicine. Why? I think because of the importance of evidence-based medicine, and that this can really have a dramatic effect on public health. I think that's why it has such a strong importance there. But you often see systematic reviews and meta-analyses commonly done, very commonly done in psychology, for example. The term meta-analysis was coined by a psychologist, Dr. Glass. And there's people are trying to be encouraged to do meta-analyses and systematic reviews, for example, in animal research and other fields as well. So although it's predominantly done in medicine, I think again, because the importance it has for public health, there is more and more interest to do it across other fields as well. As long as there's evidence that has been already been published or unpublished, you're synthesizing together in the strategy that we're going to be highlighting in this webinar, it can be done for any field, of course. Great question.
Thank you so much, Dr. Jeff. The next question, why systematic review has high bias?
I think this is probably, and thank you for asking that question, and hopefully let me clarify that maybe the confusion. I think when I said that, I was comparing it with a meta-analysis. And I was saying compared with a meta-analysis, systematic review will have a higher bias. However, let me emphasize, systematic review has much less bias than most other study types. It's just when you're comparing to a meta-analysis. And that's because a meta-analysis, you're doing a statistical synthesis. So it's a quantitative reevaluation. And because it's quantitative and there's no qualitative assessment of that information, it's going to be less biased. When it's a qualitative assessment, which is what a systematic review is, it's going to have more bias than what you would see in a meta-analysis. So yes, I don't mean that systematic reviews have high bias. In fact, good systematic reviews have very low bias. It's just compared with a meta-analysis, they'll have a higher bias because of the more qualitative nature of them. So thank you for allowing me to clarify that.
So much, Dr. Jeff. The next question is, if you get a big data from Cancer Genome website, for example, does it consider as a meta-analysis review? Or if you got a concrete idea, it could be considered as a publishable article?
It would certainly be a publishable article. It would not be a meta-analysis. A meta-analysis is aggregating together several different studies together, grouping all of that information together. But it's not just the aggregation. As we're going to see, especially in the second webinar, it's a careful evaluation of the quality of that evidence, evaluating potential biases in those different studies. How do they obtain their data? Do you have any concerns about the accuracy or the validity or the reliability of that data? And that will then influence the weight that the information will have in your meta-analysis. So that what you're describing is just a typical standard research article where the data comes from a publicly accessible database, for example, a genomic database. So that would not be considered a meta-analysis.
So much, Dr. Jeff. The next question, when can I repeat the same systematic reviews and meta-analysis which was done before?
So that's a good question. So that's, for example, maybe you have an idea for systematic review, you go into the Prospero database and you see, oh no, somebody's already doing it. It's either published or maybe it's been ongoing for a year, which means they'll probably get published a lot more quickly than you. So what can you do? How can you revise? Well, think of PICO. So think, okay, well, maybe you want to change the population of interest. Is there another population or a subgroup of population that may be relevant for? And I'll talk about that again in a moment. Or maybe the intervention, you want to vary it, or the outcomes that they're evaluating. Now, and also you may want to consider the study designs. Maybe if they did RCTs, maybe you want to look at observational studies, for example. So there's a lot of opportunities to vary what you're looking at to ensure it's going to be valuable. Now, in a published systematic review or meta-analysis, we'll be talking about subgroup analyses in the next webinar, but you may see they've done subgroup analyses where they'll see in certain groups that they saw a much stronger effect than what they saw altogether. Now, what that generally does, those subgroup analyses are really a great way to establish new hypotheses. And so if you see a systematic review and they saw a particularly strong effect in a particular subgroup, maybe want to now conduct a full systematic review specifically on that subgroup alone to really assess all of the evidence related to that subgroup to see whether or not that efficacy of that intervention is still true. So there's a variety of ways you can optimize your systematic review to avoid being duplicative or replicative of what's been done before to improve the clinical relevance of your ideas. Great question.
Thank you so much, Dr. Jeff. The next question is, how can we minimize biases in systematic reviews? What do we do if we don't have quality data for a certain population like Middle East?
So that's exactly what we're going to be discussing in the latter half of this webinar and the beginning of the next webinar. Once you've identified the studies, what's next? How do you extract that data? How do you evaluate that data? How do you assess the quality of that evidence, the biases that may be relevant? And generally what you're going to do, you're going to assign different weights. So if, for example, studies may be done in one population, if they weren't done with very strong technical rigor, yes, you'll include that data in your analyses, but because you're going to assign it a very low weight, it really won't have a strong impact on the overall effect that you see. Another study done elsewhere, which has a lot more technical rigor, larger study, a larger sample size, would likely carry a lot more weight in your qualitative synthesis, and therefore would have a more dramatic impact on your overall assessment. So I think assigning weights, again, we'll be discussing a bit later, will help you to ensure that the studies you are including are going to receive the appropriate relevance in your meta-analysis. Great question.
Thank you so much, Dr. Jeff. The next question is, what are the software for making systematic review and meta-analysis?
So another good question, and I will emphasize Springer Nature does not own any software for making systematic reviews or meta-analyses. So I'm going to recommend two. I'm going to talk about one specifically today, actually, but there's two that I would strongly encourage you to consider. One is paid for, one they do, I believe, have a free option. The one is you'll see everyone uses RevMan. Now, RevMan is a great way to be able to include all of your data, do the analyses, make your forest plots, etc. It's all automated. It's quite simple, easy to use, great tutorial, especially with Cochrane. And there's a lot of other secondary software that feed into RevMan, so it's really streamlined. The other one that I'm going to introduce, because I think it's a lot less familiar, is called Covidence. I'm going to talk about that platform today because I think it's really helpful in assessing or screening potential studies you want to explore in your meta-analyses and systematic reviews. Now, this Covidence is not free. It is something it is paid for, and I would like to, again, emphasize as a disclaimer, neither of them are owned by Springer Nature. I'm only recommending them because I think they're really useful softwares.
Thank you so much, Dr. Jeff. I think that we have went through most of the questions that we got here. Thank you for this informative Q&A session, and thank you everybody for your participation. So we can now end this Q&A session and resume our webinar. Thank you so much, Dr. Jeff.
Wonderful. Thank you so much, Heba. And for those, if we haven't gotten to your question yet, we're going to carry your questions over to the next session. So we do promise to get to them. And if we don't have a chance to get to them today, or if you have additional questions, you will see on the last slide is my email address. I do encourage you to send me an email and I'll be happy to try to help you there as well. So let's now move on. We talked about the importance of systematic reviews and setting them up in terms of the registration and identifying good questions. So let's now talk about doing the actual systematic review. Once you've established, yes, okay, this needs to be done, it's not being done already, you have a good question, it's feasible, you have the expertise, the resources, the time, etc. Now we can move forward. Now, the whole reason a systematic review is called a systematic review is that it is systematic. It means it's a carefully thought out protocol to evaluate the literature or publish or unpublished studies. It's not simply assessing the papers on your desk is simply being a much more thorough and objective evaluation. And that's what we're going to move to talk about now. How do you select the studies to include? And as you can see, I didn't say papers because they may be unpublished studies. So firstly, you need to clearly define your inclusion and exclusion criteria. So you should be looking both published and hopefully unpublished studies. What languages can you assess? Keep in mind, not everything's published in English. There's a lot of papers published in French and Chinese and German. And so if you're collaborating with people who can speak French, then you may want to include those French studies as well. Which databases are you going to use, and make sure you have a really clear rationale. So if you're going to be use double-blind RCTs as your criteria of inclusion in terms of PICOS for S, why is it so important that it needs to be double-blind? What type of biases you may be concerned about? For example, if you're looking at qualitative data in those studies or subjective analyses in those studies, having a double-blinded RCT would probably bring much greater weight or much greater technical rigor to that study than a single blind. So make sure you have a good rationale behind each of those criteria. Now, we did talk about PICOS, and this is a great place to start. So what I usually recommend is use P&I for your search strategy, and then you can use C, O, and S as for your inclusion and exclusion criteria. So you use P&I to identify the studies, and then when you're screening through the studies, you can evaluate whether or not they have the right comparator, the right outcomes, and whether or not it's the right study design of interest. In some cases, for example, Medline, you can also filter out by study design. Other databases, you don't have that opportunity. So it's generally going to be something that would be included as the screening step for a lot of your results anyway. Now, at this stage, I would recommend ask a librarian. Librarians are trained in how to do good literature searches, and so you need that training. They can help you to identify the right words, the right Boolean operators, help you with controlled vocabulary. So Medline uses controlled vocabulary, which help you to search within, they call the MESH, the medical subject headings. But for example, Web of Science does not use a controlled vocabulary. Most of us are not trained in library sciences. We're not familiar with all that information. So working with a librarian can really ensure you're developing the right search string to identify the best studies. Keep in mind synonyms in your terms of your search strategy. So if you're looking at, for example, taste, you may not just want to use flavor, but use both flavor and taste in your search terms. You can also use Boolean operators like and, or, not to really help to narrow down the results so you're not getting too many that you have to screen through. You can use truncation symbols. So by putting an asterisk, for example, after child, that would pull back anything as child, children, childhood, anything that begins with child. So that helps you to be a lot more inclusive in your analysis. And then you can use also proximity operators. So for example, if you use ADJ, which stands for adjacent, and you say fracture ADJ foot, basically when it's looking for the literature, it's going to look anytime where the word fracture and the word foot are close by. They don't have to be next to each other, but just within 1 or 2 words of each other. So for example, fracture of the foot and foot fracture would both be identified by using this type of search string. So again, most of us are not familiar with how to do this really well, so I do encourage you work with a librarian. Now, in terms of describing a search strategy, I mentioned to you that a lot of what we're going to be discussing today will be coming from an editorial reviewer perspective. So what are you expected to explain in your papers related to your search strategy? Is A, should you highlight the databases that you searched in? B, should you list all of the articles that you screened? C, should you share the search strings that you use to identify those studies? And or D, should you state the dates when those searches were conducted? So now we're going to launch poll number 4. You can select one or more of these options.
So much, Dr. Jeff. So everyone, now you should see the poll on your screen. So please choose one or more of the 4 options and submit your vote to us, and I will keep the poll open for a minute to give you time to vote. So how to describe your research strategy in your paper? Is it to indicate the databases you searched, or provide a list of all articles that were screened, or share the search strings that were used, or state the dates when the searches were conducted? so much everyone for your participation. So I will now close the poll and I'm going to share the results on the screen.
Okay, so I can see the most popular option was indicate the databases you searched. That's definitely true. Second most popular, state the dates when the searches were conducted. That's also very true. 56% said provide a list of all of the articles that were screened. So maybe I think some of you who have not yet conducted a systematic review, you'll be maybe surprised to know that you're likely going to screen through about 500 or 600 articles. And if you were to list all 500 or 600 articles, we can imagine how many pages long that would be. So you do need to emphasize or list as your references the ones that are included in the study, but the ones in the initial screening, you don't need to include, because again, it would generally be within the hundreds. And I can see only 36% said share their search strings. Well, actually, you should share their search strings. That's the importance of transparency, so people can validate whether or not how you identify that literature was appropriate or not. So it'd be options 1, 3, and 4 are all components of a good systematic review paper. Thank you very much, Heba. So let me show you an example of what I mean by the search string. So only 1/3 of you think that's relevant, but let me just show you how you'd actually share that. So here's a paper looking at the benefits and adverse events of ACE inhibitors in patients with heart failure with reduced ejection failure. So again, this was a nicely prepared systematic review. So here's PECOS. They're looking at patients or adults with reduced ejection fraction heart failure. Their intervention were ACE inhibitors. Comparison was placebo. They identified the inclusion exclusion criteria, so what benefits they're looking at, what risks they wanted to evaluate, and for the study, they only looked at double-blind RCTs. Now, as I mentioned, generally for the search strategy, P and I is what you'll use. CO and S are often usually done during the screening step, not the selecting step. So let me show you the search strings they used that they published with the paper. They published it as additional information, supplementary information, but it's transparently available to really highlight the patients as well as the intervention. So they looked at Central, Embase, and Medline. We'll talk about those different databases in a moment. For Central, so in blue, what they have listed are related to the population, and then in I is what's related to the intervention, ACE inhibitors. For Embase, again in blue, they have their population, and then in red, they have their intervention, the ACE inhibitors. So you can see they were very clear. They had two different search strategies, different databases do require different search strategies, and they were transparently shared that. Well, what about Medline? Medline is a lot more complicated. Here's the search strategy they use for Medline, which again is published as supplementary information. In red, they have their information related to the intervention, ACE inhibitors. In blue, they have the search items related to the population of interest. And then they also mention down in black, you can say they're looking both at English and French language. They wanted to only look at adults, so they don't want children, and they don't want any animal studies. So this is what I said. Most of us would not be able to write a search string like this without a librarian's help. So I do encourage you, you are expected to publish your search string with your systematic review as supplementary information. And when reviewers see that your search is not appropriate to identify the relevant literature, that would reflect poorly on the evaluation of your paper. So again, work with a librarian from the beginning. So what databases should you look at? Well, as I mentioned, you should always be looking at both published and unpublished literature. So published literature is often referred to as the white literature. So you can look at, for example, PubMed, Cochrane, Embase, etc. This is looking at all those journals that have been published. But there's also the gray literature. This is unpublished studies, often conferences, theses, etc. that you can then get additional information from. And especially if you're looking at RCTs, you should always be looking through clinical trial registries as well, because a lot of clinical trials do not get published, and therefore they would be excluded from your systematic review. So although you're probably familiar with PubMed and Embase, You may not be familiar with the Cochrane Library. Again, Cochrane is an excellent resource for systematic reviews, and their database is really useful as well. So theirs is called Central, and it does draw from both PubMed, Embase, as well as clinical trial platforms. So it's much more inclusive than just looking just only at PubMed or just an Embase. And for example, this is when I visited yesterday, they have 1.7 million trials currently in their database that you can freely access to get that information on. So for example, I put in hepatocellular carcinoma, and I can see there's over 4,600 trials currently in their database. You can then filter them by year that they're published in. You could also see where they're actually taken from. So you can see if they're actually published or unpublished studies, for example. Now, another database you may not be familiar with, but I think can be useful, is ATIS Insight. As a disclaimer, this is one of the few databases I'll recommend that is owned by Springer Nature, but only because it's a bit different. It's a pharmacological-based database. So if you really want to look at a specific drug, then ATIS can be really useful. So basically, just type in your drug name and click on search. So in this case, I looked at sorafenib, and they can see there's over 1,000 trials in their database. And one of the reasons I like ATIS Insights, why I think it can be valuable, is you can also filter by what phase. Is it a phase 3 trial, phase 2 trial, phase 1 trial? And you can also check the status of that trial. Is it completed? Is it ongoing, etc. So I think having, again, a little bit more refinement in terms of the trials, but just keep in mind, they will be focused on pharmacological interventions only. Now, of course, you can also contact the authors. And I do recommend, keep in mind, in even published RCTs, a lot of information related to that study may not be published in that study. The author may thought it wasn't necessary to include, but that may be relevant information for you to be able to get from them. So all those studies you have decided, maybe you decided to have these 8 RCTs in your systematic review or a meta-analysis, I would strongly encourage you to contact those authors, ask them if there's a different additional information they can provide, additional data that's not published in that paper. And for any paper, when you go to the corresponding author, you will always find the email address of that author, so it's easy to contact them. And if you're in clinicaltrials.gov, for example, which may be an unpublished paper, and again, if it's unpublished, they're supposed to upload all the results to the trial registry. Doesn't mean it's updated. I would still encourage you to contact the author. And again, it's always easy to do so. They will give you the email address of the person who registered this trial. So I do encourage you to contact the authors in addition to simply screening the papers to really make sure it's a much more complete and rigorous systematic review. So let me show you a nice example of a published paper where they really describe their search strategy in careful detail, and you can see the thoroughness that they use to try to identify it. So this is one I introduced earlier, the efficacy, effectiveness, and safety of vaccination against HPV in males. So first, they emphasized in the methodology that they had registered this in Prospero, and they give their registration number, and that they also followed the PRISMA statement to ensure they're adhering to the international guidelines. Now they want to talk about PICOS. So they first introduced the population intervention and comparison, so PINC. So they said they wanted to investigate efficacy, effectiveness, and or safety of vaccination with a vaccine against HPV in males of any age. So they identified the population as well as the intervention. And they mentioned the control group had to be males who were either unvaccinated or had received placebo or vaccine not directed against HPV. So they clearly identified the comparison as well. And then in terms of outcomes, they now go on to list 12 different outcomes that they wanted to assess from those different studies, being very clear of what they're going to be. Now, in addition to highlighting their established research question using PICO, they also go on to emphasize the databases that they use. So they use Medline, Embase, and Central from Cochrane. Those are the 3 standard ones that almost everybody uses. They also say when they did their initial search, which was on November 4th, 2016, but they also did the latest update to that search on April 18th, 2017. This is something you would never see in a literature review. And they say if you want details on the complete search strategy, look at the supplementary information. This is where they will provide the search strings. That's always going to be an expectation. Then they say, additionally, they also looked at clinicaltrials.gov to search systematically for unpublished or ongoing trials. They're being more thorough. Now, in addition to looking at these online databases and contacting authors, something we talked about already, can you think of any additional strategies to maybe identify other studies that you were unable to identify in these online databases? Any other strategies you can think of? Please write them into the questions box. So besides looking at like PubMed and clinicaltrials.gov, what other ways can you try to find studies? Any ideas? Okay, so I'm gonna say preprints. Yeah, preprints is quite common. Web of Science is actually a database, and I'd say preprints is actually a database as well. Cochrane database is a database. Government policies, very good. Look at government policies. Some say conferences, exactly. Drug banks, dissertations, a lot of fantastic ideas. And if you do that, describe it. And that's exactly what they go on to do. They said they'd also performed electronic searches were complemented by manually searching conference abstract books. So they took the books and they read through them of major international HB conferences. And they highlighted the 2 conferences that were relevant, as well as reference list of all the identified studies. Once they had identified studies, they then looked through the reference list of those identified studies to see if they can identify any more that wasn't identified in the original search. So a manual search is often thought to be even more thorough than simply relying on databases. And again, transparently described. And then is it one person? No, as I mentioned, systematic reviews are almost always need a minimum 2, usually 3 reviewers. And why? Because everything has to be done in duplicate. So they mentioned that the search results, the titles, abstracts, and full text were independently assessed by 2 investigators, and they highlight those 2 investigators. These are the authors of the paper, and differences were discussed until consensus was reached. So again, you can see a very thorough description of their search strategy. So when you are planning your strategy, make sure it is thorough like this, but you also have to describe it in the paper as well. Additional information, they said, we did not make any restrictions regarding setting, language, or publication status. However, potential indirect effects of male HPV vaccination on the incidence of clinical outcomes in females was not considered. So again, they're giving additional information that reviewers probably asked for. Now, when you're describing this, you also not just do it in text, but you also need to do it with a flow chart. And it's usually recommended to use the Prisma flow chart, which you can download from the Prisma site that I mentioned to you earlier for free. And it tells you exactly what information you should include. Let me show you an example of a nicely prepared Prisma flow chart showing how they identified the selected studies. So here they identified 629 papers or records. Can you imagine listing all 629? No, you don't need to do that. And they said 356 from Medline, 225 from Embase, etc. So they describe exactly where they're actually found from. And they also found 7 additional records as well outside of that, probably from manual searches. And then from that, they screened through them. And from all of those records, they found 44 that they thought were going to be appropriate for the inclusion exclusion criteria. They then evaluated those 44 papers for full text. So looking at methods, results, the figures, the discussion to make sure they are in fact appropriate. If not, they were excluded. If they are excluded, they have to emphasize why. So they said they realized 7 were not RCTs. They thought they were, but they weren't. 5 didn't meet exclusion criteria. 4 were actually reviews. 1, they didn't get an answer from the authors, probably unpublished study, etc. So they ended up having 25 articles which had 6 primary RCTs, and of these 6 primary RCTs, they then used 5 of them in their quantitative analysis, so the meta-analysis. So you can see it's a very carefully laid out description of how the search strategy was performed. And this is what would be expected of you when you are preparing your papers describing your systematic reviews. Now, someone had asked earlier about any databases I might recommend, and I said one that you may not be familiar with, but I think it is really valuable, is called Covidence. And again, as I emphasize, has no association with Springer Nature. I just think it's a good database. It's not free, but if you do conduct systematic reviews, that it may be something worth investing into. And they do have discounts for people in emerging markets like Egypt. Egypt was one of the countries that they said if you are in Egypt, you can contact them to get a potential discount on the actual subscription fee. One of the benefits of Covidence, it does feed the information directly into RevMan, which is what I mentioned people often use to do their analyses and prepare the forest plots. So it helps you to have a nice streamlined procedure. So what does it help you do? Well, you can import papers, firstly. Secondly, in there, the database alone, you can then screen titles and abstracts, screen the full text. You can assess the quality of the evidence provided, then extract the data. Then the extracted data can then be fed into RevMan for the data analysis. So again, nice systematic workflow. So the purpose of this webinar, I'm just going to talk about briefly about screening titles and abstracts and screening the full text. Because once you've selected all those studies, the next step is to go through, read through all of the titles, all of the abstracts to see, is it appropriate, yes or no? If it is, it moves on to the full text screening. Is it appropriate, yes or no? And so one of the things I like about Covidence, it makes it automated. So it's really simple. So you log in, again, you have two investigators. They would both log in and they would have the list of all the titles, all the abstracts of all the studies they identified. And then based on reading the title and abstract, they can vote yes or no. Maybe is basically a yes. And yes means it's then moved, it's done. And then they're automatically shown the next one, yes or no. So it's nice and automated. Then the benefit is you can then track where you're at. So for example, what we're showing on this screen here, so there's 9 studies that have already been done by both investigators. And of those 9, one of them, there's a conflict, which means one said yes, one said no. So now they have to resolve that conflict before it can move on. So that means 8 have already been agreed upon, and you can see down here, 8 of them have already been moved to the full text screening. So it's a nice automated procedure, which I think makes things easy. You can see where you're at, where your partner is at, your colleague is at, how many have not yet been done yet, and I think having that makes it a lot easier for you to manage. The next step is the full text. This will take time. You have to read through the entire paper, especially the methodology, figures, and results and discussions to really ensure about the methodological rigor, that it's appropriate, it's the right interventions, outcomes, etc. If not, you have to exclude it. If you think it is appropriate, you simply click include, and then it'll be included. Again, a nice, simple, automated workflow, which is why I recommend this is a very useful database to use. Same with the title and abstract screening. You have the same idea with the full text. You can see how many you've actually done, how many have been agreed upon, how many conflicts that you have. And then if once both investigators agree, yes, this is a full text, this is something we should include, it will then move on to data extraction, which means we have now one study that both people have agreed upon that's now moved on to the data extraction. And so now the authors can go through to just extract that data. We'll talk more about data extraction in the next webinar. So with that, let's now move on to activity 2. And activity 2 is going to be focused on selecting studies. And this is found on page 9 of your workbook. So for activity 2, so during your literature search, you identified 324 RCTs in Medline and Web of Science databases, together with 4 unpublished studies from conference proceedings. And although the published RCTs all have an English abstract, 34 of those papers are actually in French, Spanish, or in Japanese. How would you narrow down the final studies to include in your meta-analysis? Would you A, since no research on your team speaks French, Spanish, or Japanese, remove the 34 foreign language studies from 324, instead just use the 290? B, remove all the duplicate studies from this 324. Consider adding the 4 studies if all the necessary data and methodology are available to unpublished studies. Keep the 21 French studies that can be understood because you do have 2 French-speaking colleagues who can probably help you to assess those studies. But you would remove the 8 Spanish and 5 Japanese studies. Or C, remove all the duplicate studies from the 324 RCTs, add the 4 published, unpublished studies as long as all the necessary data is available, and you would actually keep the 34 foreign language studies because at least the abstracts are written in English. So which of these options would you choose? So Heba, can we now launch poll number 5 for everyone?
Thank you so much, Dr. Jeff. So everyone, now you should see the poll on your screen. So please choose one or more of the options and submit your vote to us. And I will keep the poll open for a minute to give you time to vote. So thank you everyone for your participation. So I will now close the poll and I'm going to share the results on the screen.
Okay, so I can see the most popular option was option C, to remove the duplicates but keep all the foreign language studies. Okay, thank you very much, Heba. Well, if I was going to advise these reviewers, I would recommend actually option B. Now we'll talk about the benefits and the conditions or limitations of each. For A, first of all, one issues, there's what about the duplicates? So it's fine if you want to remove studies, that's understandable, but please be sure to always remove duplicates. So that's why I'm not surprised to see that most people did not choose option A. And there will be a lot of duplicates. A lot of papers in Embase are also in PubMed as well. And then option B, this is really the best approach. Why? Because you're removing the duplicates. You're going to use the 4 unpublished studies as long as you can get all the methodology and information from those authors. But if you can include the foreign studies, you have to have people that can read all of the paper, not just the abstract, but the entire paper. And you need to have at least 2 people, because remember, you need 2 independent assessors. So what I would do in this case, I would recommend those 2 French-speaking colleagues would be collaborators onto the systematic review, and they would be the assessors of the specific French studies in the systematic review. But for the Spanish and Japanese studies, because you cannot assess the data or the actual methodology directly, you would have to exclude them. Now for C, although this was popular, keep in mind that the abstract is not providing you enough information to really understand thoroughly the technical rigor, the study design, the analyses, limitations, objectivity, all of that information which will be essential when you're trying to evaluate the potential biases in those studies, which is something we'll talk about in the next webinar. So the last thing I want to talk about before we conclude this webinar, however, we still have a little bit more content to cover and won't be much, but it's about the data extraction. So you've screened through those studies, you've identified the full text, these meet the inclusion-exclusion criteria appropriate for PICOS, and now it moves on to the data extraction. Now, in data extraction, there's a couple of different ways you can do so, but what I always recommend is to use a standardized, formalized data extraction form. Easiest one is from Cochrane Collaboration. As usual, they're freely available. You can simply download the data collection form, and this will give you for both RCTs or observational studies. And you can see, it'll tell you, here's the information about the general information, characteristics of the included studies you should include, information about your risk of bias assessments. Again, we'll talk about in a bit. Talk about the interventional groups included in that study. It's also going to talk about other information that may be relevant. So several pages, you can simply use the text boxes you can fill in. You'd have two independent investigators, both fill them in independently, compare the results to see if they agree. And if they don't agree, generally a third person will come in to try to resolve those conflicts. So that's why minimum, usually you need 3 reviewers for a systematic review. But covidence also I think is quite useful. I want to emphasize that I do understand this picture is very blurry. I simply took this as a screenshot from their YouTube promotional video because that's all the information I could get from that. But again, one of the things I wanted to emphasize about covidence, it is customizable, something that Cochrane one is not. So you can simply say, okay, I'm looking at dichotomous outcomes, and then based on dichotomous outcomes, here are the ways that they can be reported. So odds ratios, risk ratios, etc. So it's a much more customizable approach than the Cochrane data extraction form, and that could be quite useful. But again, 2 independent reviewers do it. They can then compare. Any conflicts would then be resolved before moving on. Now, what type of data should you be extracting? Well, there's a variety of different data you're likely going to encounter in those studies. You may be seeing continuous data where there's consistent intervals, for example, like blood pressure. And they can be either, for example, they can be they can be interval or not. And if you have continuous outcomes, then what you're going to be extracting in terms of how you're going to compare them across studies depends on the scale that was being used. If all the studies use the same scale to evaluate that same outcome, then you can just simply use the mean difference between those studies. However, if they use different scales, which may often happen, or at least even one of the studies uses a different scale, then what you should be using is the standardized mean difference, which is basically you take this mean difference, you divide by the standard deviation in that study, that gives you the standardized mean difference. And therefore the standardized mean difference helps to normalize a study, means you can now compare them to other studies as well. In addition to continuous outcomes, there could be dichotomous outcomes, so binary, so yes or no, alive or dead. And these are usually going to have odds ratios, risk ratios, numbers needed to treat, absolute risk, etc. These are probably very commonly going to be used. One of the benefits with these types of association measures is that you don't have to worry if it was use the same scale or different scales because they are associations. You can just go ahead straight across. You compare the risk ratios, the odds ratios of the different studies. However, you compare like with like. So you don't compare risk ratio with an odds ratio. You compare risk ratio to another risk ratio. So if some studies looked at odds ratios, some studies looked at risk ratios, they should be compared separately from one another. You don't pool those together. You may also want to look at time to event. So for example, survival time or time to remission. Now this is a little bit more difficult to really do in a systematic review. What people do more commonly is they'll try to dichotomize this information. So if you have, for example, let's say at the 12-month time point, you have the information for all the participants in those studies, and you can then calculate, okay, in all of these studies, how many experienced an event within the first 12 months. And therefore, you can then have your table that you can prepare, the 2x2 table, and with your contingency table, you can then easily calculate the risk ratio or odds ratio, therefore allowing you to dichotomize these time to event measures. Now, the other types of measures you may see are ordinal, like Likert scale, from people experience pain, no pain to a lot of pain. So those types of rankings or ordered categories are not frequently used in systematic reviews, nor are counts to event, and usually infrequent events. So the number of myocardial infarctions or the number of strokes that people had received. It's not the time to an event, it's how many times they experienced that event. And generally those are not going to be often used either. So more commonly, continuous dichotomous outcomes or times to event, but dichotomized to make it easier to evaluate. So how do you compare? How do you emphasize and describe that data? What are reviewers, what are editors looking for? So in this paper here, for example, they wanted to describe how they extracted all the data and what data was extracted. So they go on to say that the following records were extracted to include the study for the study reviewers by the same 2 study reviewers, and then they then go on to highlight all of this different data that was extracted from those different studies. So being very transparent. And then they emphasized the reviewers, the authors that were involved. They said the 2 reviewers independently abstracted the data, and 2 other authors, who they listed their initials here, resolved the inconsistencies. So again, from an editorial and from a reviewer's perspective, it all comes down to transparency and exactly describing how the systematic review, in this case meta-analyses, are going to be done. And the last thing I want to talk about, we touched upon this in the Q&A session, is about weight. Because yes, different studies will have different qualities. Large studies generally will have much higher precision, and therefore smaller standard errors, and therefore less variance. And therefore generally thought to be seen to have stronger weight. But it's not just the size of the study, it's also the methodological rigor of the studies as well that would influence it. And so generally the weight that's applied, how it's done so, it's basically the inverse of the variance. So therefore large studies that are going to have much less variance will have much stronger weights than smaller studies that are going to have much higher variance. And then that will then influence on how you're going to do the statistical synthesis of that data in your meta-analysis. How would that be reported? Well, in this paper here, looking at this GATM polymorphism of statin-induced myopathy. So again, for meta-analysis, usually reported as a forest plot, in which you can see they first highlight all the odds ratios, but in addition to the odds ratios of these 6 studies, they also emphasize the weights of the studies. You can see the weights equal 100, because they should equal 100, but what percent of this total value came from these different studies? And that's going to be dependent on the size of the studies, as well as the methodological rigor. So you can see, for example, this one had the highest weight. You can see it had a lot of participants in the study. The one by Seidel had very little weight in the meta-analysis. Why? Because it's the smallest study that was included in the meta-analysis as well. So all of this information needs to be transparently presented and described, and therefore reviewers and editors feel that the information you are providing will have stronger benefits for the clinical population of interest. So with that, that brings us to the end of our first webinar. We're now going to move on to our Q&A session. So we've had a few questions come in from previous already, and we encourage you, if you have additional questions, please write them into the questions box. We'll get through as many as we can before we conclude today's webinar. So with that, Heba, can you please read out some of the questions that have come in already from the participants previously?
Jeff, the first question we got here, what's the minimum number of papers or studies that should be included to conduct a valuable systematic review?
Great question. We're going to talk in a bit more detail in the second webinar when we talk about publication biases, but generally I'll tell you now, it's about 10. Basically, if you have less than 10, it's difficult to assess potential publication bias in your systematic review. If you have 10 studies or more, you can feel more confident you're more inclusive. Good question.
Much, Dr. Jeff. Another question here is, just a moment. Can an undergraduate write a literature review by his own? If yes, how can he qualify himself to do so in an international journal?
So unfortunately, this webinar is not about literature reviews, and we do have a lot of questions about systematic reviews, so we don't have time to address that question. But if you want to write to me separately, I'll be happy to help you with that.
Thank you so much, Dr. Jeff. Another question here is, what's the minimum number of papers or studies that should be included to conduct a valuable systematic review?
So we just answered that one, and it was 10. So maybe that's the same person wrote the same question twice, perhaps.
Okay, so another question here is, to write systematic review, should I have strong biostatistic knowledge, or can I go with basic statistics? I mean,
I would
should I have the skill to evaluate the statistical method used?
strongly encourage you to work, if you do not have a strong statistics background, I would strongly encourage you to work closely with a biostatistician when organizing the study protocol. and making sure you're doing it properly, especially if you're going to do a meta-analysis, which can be quite complicated in the statistics. I would strongly encourage you to work closely with a statistician. If you're just doing a systematic review, the qualitative synthesis, maybe your basic statistical background would be sufficient. But if you're going to do a meta-analysis, I would strongly encourage to consult with a statistician. They always know best.
Thank you so much, Dr. Jeff. Another question here is, can I know the exact difference between the systematic review and the meta-analysis?
So great question. So systematic review, I'd say a meta-analysis is an extension of a systematic review. They're always systematic reviews, but then of a systematic review, sometimes you may feel it's going to be worth to actually pool that data together, reevaluate it statistically to actually present this kind of forest plot. This is a meta-analysis. What they did, they pulled all the data from these 6 previous studies, reanalyzed it statistically, and then they came up with this diamond. What this diamond is showing is the overall effect of all of these different studies. The width of the diamond is basically the confidence interval. And because it does not cross the threshold, we can now say that yes, we do think that there's going to be an association with this polymorphism with the actual condition of interest. Systematic review is a qualitative synthesis. It's basically doing everything except pulling that data together. And in the next webinar at the end, we're going to talk about doing the qualitative synthesis and doing the statistical synthesis, and you'll see some of the differences involved and actually how they're going to be performed. But again, to emphasize, meta-analyses always include a systematic review. Systematic review does not always include a meta-analysis.
Another question here, how to conduct meta-analysis if the studies shows heterogeneity?
So now we're going to be talking more about heterogeneity at the second half of the next webinar. We're going to be talking about the fixed effect and random effects models. Random effects models going to be more appropriate when you do have heterogeneity across the different studies. We'll talk a little bit more about heterogeneity in the next webinar. But if you do feel it is going to be appropriate to pull that data together, you are concerned about the heterogeneity across those studies, then you should be performing the meta-analysis with a random effects model.
Thank you so much, Dr. Jeff. Another question here. If I'm interested in certain drug or disease and I don't have a nearby fellow, can I find 2 or 3 since it's difficult for me to work alone?
I think maybe find 2 or 3 collaborators, I'm assuming, and I would say definitely, as I mentioned, systematic review minimum has to be done by 2 people. One person cannot do a systematic review. Well, they can do it, it just will never get published. You need a minimum of 2, usually 3. And that's because 2 independent people need to do the selecting the literature, screening the literature, abstracting the data, evaluating the biases, etc. And then a third person is usually necessary then to resolve any potential conflicts between those 2 investigators. So yes, if you are by yourself, I'd encourage you, the benefit of systematic review, you can do them long distance because you're simply assessing information on your computer. So it's much easier to do when you can't be working adjacent with a colleague face to face.
Thank you so much, Dr. Jeff. Can we use a single database as Cochrane database or Covidence?
So Confidence is not a database, it's a software to help you to do the screening and extracting. That's really all it is. It's not a database, it's just a software you could use. It's like EndNote to help you manage your references. Covidence is a software to help you manage your selecting, screening, and extracting procedures. So don't confuse Covidence with an actual database. So Cochrane, Central from Cochrane, Embase, PubMed. Yes, you can just use one. You'll probably have difficulty get it published. Unfortunately, the expectation is you'd always use multiple databases. As I mentioned, almost all good systematic reviews will always do Medline, Central, and Embase. In addition to that, we'll also look at the clinicaltrials.gov. I would say that would be probably the 4 minimum databases you should be using in your systematic review and meta-analyses.
Thank you so much, Dr. Jeff. I think that we have went through most of the questions that we got here. So thank you so much for this informative Q&A session, and thank you everyone for your participation. We can now notify our audience about the last notices we got here in the morning webinar. Thank you so much, Dr. Jeff, for this amazing webinar.
And thank you very much, Heba, for your wonderful facilitation. I'd like to thank all the participants for the great participation and great questions and interacting with us on all the different activities throughout this morning. We do hope so far the webinar has been useful. We'll be coming back in an hour. In the next hour, we'll be talking about assessing the quality of evidence, what kind of biases you should be looking at, how do you report those biases in your systematic review, potential biases in your systematic review, what we call meta biases, you have to be careful of to really ensure that your papers get through the review process more smoothly. And finally, we'll compare and contrast qualitative and quantitative syntheses with the systematic reviews and meta-analyses to conclude the actual next webinar. So with that, thank you very much, everyone. Have a great break, and we'll see you again shortly. Thank you.
So much, Dr. Jeff, and we can now end the webinar.